Hey, good morning, everybody. As you heard, I'm Barry Canton. I'm one of the co-founders and the CTO of Ginkgo Bioworks, a biotechnology company based here in Boston. I want to start off by thanking Google for inviting us to be here today. It's great to have an opportunity to talk about our company a little bit, talk about how we're using AI and some of the impact that we imagine that it can have for us and for the industry more broadly. As Joe mentioned, we recently announced a major cloud and AI partnership with Google, and we're happy that we have pressed the easy button for Google Cloud and AI. So I'll jump into it. I've been doing this for 20 years, and Ginkgo has been around for 15 years. So happy 25th birthday to Google. Ginkgo, we just had our 15th birthday recently. I would say that there have been a very small number of technologies that have emerged that we collectively believe can have a very major impact in how we do the kind of work that we do. And I think that the Gen AI is very clearly one of those, and we're excited to be able to leverage it and talk about it today with all of you. First about Ginkgo. Ginkgo is a company whose mission is to make biology easier to engineer, and that's why I'm so interested in technologies that can move that forward. Our business is to be a horizontal platform for programming cells, and we do that across a range of application areas. And over the course of the talk, I'll unpack that somewhat cryptic statement. As I said, we've been around for about 15 years. We leverage robotics, we leverage software, we leverage biological tools, wetware, if you will. And we have an incredible team of engineers, technologists, biologists, scientists, and the full spectrum of disciplines needed to do this kind of work. All right, so let's kind of unpack what I mean by a horizontal platform for cell programming a little bit further. Well, one aspect of being a horizontal platform is that we need to be able to go end-to-end with scientific projects, where end-to-end might mean starting with computational discovery of a protein, an enzyme, a pathway. Then it moves on to optimization of that molecule or protein or pathway or whatever it might be, using high-throughput experimentation to make sure that we have identified the best possible product. And then it moves on to optimizing and developing the manufacturing of whatever that product might be. That's typically, in our case, that is some kind of precision fermentation or cell culture process, followed by some kind of extraction to purify the compound or the protein that you're interested in. And at Ginkgo, we've had to build the capabilities to be able to do all of those things from end to end in order to support our partners. Speaking of our partners, and this is the other aspect of being a platform company, is that we don't actually develop and launch and commercialize our own products. Instead, we're a platform that helps our partners do that. And as you can see here on this slide, our partners kind of run the gamut from smaller companies at the bottom, larger companies at the top, and then a wide range of market verticals from pharma to industrial biotechnology to agriculture. And being a platform means that we want to be able to serve as many customers at as many different scales and in as many different market verticals as possible, and help them bring their products to market. Being able to work across that whole range of markets and contexts has required us to build a lot of different kinds of technologies. But the underlying reason why we are able to build a business that serves all of these different companies in different markets and build a platform to do that is for one sort of fundamental reason, which is that life was invented once. All of life, regardless of the kind of cell you're talking about, runs on the same underlying DNA code. That code is fundamentally programmable like any other code. We are getting increasingly good at reading that code, understanding it, and then writing new code that does something new. And the core hypothesis of Ginkgo is that since all of life runs on essentially the same code, if you get really good at programming or manipulating that genetic code in one context, you can apply that in other contexts, and you can be good at it there as well. So expertise in working with a bacteria translates into expertise in working with a yeast cell, translates into expertise working with a mammalian cell. and that expertise is partially, but not all that dependent on the particular product or market area that you're going after. It's all just reading, writing DNA, and thinking about what cells do. Now, we often like to say at Ginkgo that we can program cells just like we can program biology, just like we can program computers. But there are some big differences that we need to be aware of. Biology today is programmable, but it is not predictable. We don't always or often understand what the impact of a particular change will be in a cell that we're working with. And for that reason, when we're programming biology, there is a computational design creative component to it, but there is also a highly empirical component to it, where we need to regularly go into the lab, run often very large experiments using automated platforms like you see here, use those experiments to inform you about the biology that you're working with, and then go back to the computer and do another round of design and learning, and then iterate through that loop multiple times. As I think you'll see, that paradigm is very close to the paradigm that is being used today in machine learning and artificial intelligence, where you model, you train using real data, and you iterate until you have a state-of-the-art performance. We're very excited about plugging generative AI into this loop increasingly in our operations to make us better, and I'll talk more about that in a little bit. We're not, obviously we're not the only people seeing this. This is a quote from one of our partners, Novo Nordisk, and Brian Vandal, who's their head of global research technologies. And you can read it yourself here, but what he's saying is there is a revolution happening. It is a revolution that involves collecting large multimodal data sets and using those data sets to train AI models and using that to accelerate drug discovery, drug development, and a whole host of other applications. And we are excited to be playing a role in helping Novo and all of our partners to do that. So I'm going to round out a little bit more of the story about the Ginkgo platform, and then we'll get deeper into the AI part of the story. So you might ask, well, why is Ginkgo interested in working with all those partners in different markets? Why not kind of pick something and focus? And a big part of the reason is that there are economies of scale. So both physical economies of scale, learning economies, that the more work that we do, the more programs we work on, the more partners we support, the larger the scale we can run at, the faster we can learn, and everything gets better for all of our partners. Not just for us, but we're able to offer better experimentation, larger experiments, better data to our partners to accelerate their projects. And in bringing generative AI and Google Cloud into that loop where we iterate between experimentation in our what we call our foundry, learning, which we call our code base, and then adding AI on top of that is really just a tremendously exciting accelerant that we are pushing on very hard. To talk a little bit about scale of data, as you'll see, there are lots of different kinds of data that we care about, but one of the most important, as you might imagine, is DNA and natural DNA. So we benefit from the fact that biology has been evolving for an extremely long time and has been optimizing itself for an extremely long time, and we can just go out into nature, go out into the environment, read that highly evolved and optimized DNA code, and hopefully learn from it. And so we have been doing that at scale. What we're comparing here is the size of some of the most relevant protein databases that are publicly available in blue to Ginkgo's proprietary library in green, and how those have been growing over the course of the last 5 years or so. So today we have an extremely large, what's called a metagenomic database of approximately 2 billion unique genes that are all nature's code optimized for different purposes in different niches. And we can access that code to learn from it and hopefully find DNA sequences that are useful in a variety of different projects. So that's kind of going out and reading nature's code. There's also the question of, well, what does it do? Because it's not clear what that DNA code does simply from reading it. So the other thing we do at scale is we run high throughput and how particular pieces of DNA are performing under different contexts. And the illustration here is just showing the scale of characterization work or screening work that we have done in particular classes of enzymes. So each row corresponds to a different type of enzyme. The size of the bars indicate how many thousands of enzymes we've looked at in that particular class. And the different colors in the horizontally stacked bars tell you how many different programs, commercial programs, in which we have looked at that particular class of enzymes. So what this is showing is across many programs, we are looking at many enzymes, but the same enzyme classes show up in many programs, which, as you can imagine, indicates how there's a kind of an economy of scale around learning and around doing this work across many programs. It informs how you're going to pursue it for others. And there's some statistics on the bottom of kind of the scale of the number of programs that we've run, the number of campaigns we've looked at. I'm certainly happy to kind of go into more detail on that for folks who are interested. So that's one particular class of data. As you heard from Scott, as he laid out very nicely, in the real world, you typically care about different kinds of data, and you need to be able to collect different kinds of data. And so the platform that we've been building, which again consists of two primary components, a foundry for generating data, and a code base, which is a collection of data assets that we have built up. Within those two kind of pillars of our platform, we have a collection of assets and resources for both generating different kinds of data at scale, and then collecting that data, curating it, archiving it, making it available for our future programs. And the inherent in working with biology is a need to collect different kinds of data. You know, as shown here, it could be next generation sequencing data, it could be mass spectrometry data, it could be fermentation data. You need to be able to collect all of that to answer relatively complicated questions that come up in biology. And just to emphasize that point, if you have, say at Ginkgo, a whole category of different data types as shown on the left here. That could be nucleotide level data. It could be image level data, high content imaging for phenomics studies. It can be cell-based assays. You have those collections of data sets and data generation capabilities. And then on the right, you have a collection of applications that you're interested in going after. Any single one of those applications, I'm highlighting gene therapy here, is going to pull on a subset but multiple different biological data types that you need to be able to integrate together into a combined understanding of what's relevant to that particular application area in order to make good decisions and be able to deliver a highly optimized protein or strain or compound to a partner. And so you pick a different application area, and it's going to pull on a different collection of data types, and we need to be able to support them all. Maybe it's target discovery, and you need to be able to pull very heavily on high-throughput phenotypic screens, high-content imaging in order to understand what genetic changes are driving phenotypic changes. Okay, so that's kind of the overview of the Ginkgo platform. So now for the second half of the talk, I want to focus on AI for Ginkgo, our partnership with Google, and then how we use that in one particular, how we use AI in one particular domain today. So what we have been excited about is finding in Google a partner with equivalent ambition to us. And in fact, over the 15 years of Ginkgo, we've often looked to Google as a model for the kind of business that you can build, and the power of ambition to, in Google's case, organize the world's information. Our equivalently ambitious mission is to make biology easy to engineer. And so all our interactions with Google at a technical, a commercial, an engineering level, a scientific level, there's just been a really great resonance in ambition, in culture, in caring about what we do. And as a platform company, we understand the value of platforms. We understand the value of the Vertex AI platform. And so it's just very natural for us to adopt a platform like that and use it to accelerate what we're doing. On the AI side, as our VP of AI enablement likes to say, by virtue of partnering with Google, we now have effectively unlimited compute available to us. We are no longer bandwidth limited by compute. It's just a question of how good is our data, how much of it can we collect, and how fast can we build and train useful models. But this is really exciting partnership from our perspective. This may be very obvious for some of you, but I do want to kind of draw an explicit analogy between natural language and the language of life. So on the top here, you have this kind of progression of letters to words to sentences to documents. And as everybody knows, at this point, the transformer-based large language models and foundation models are becoming extremely good at helping us to work with that kind of natural language. Below, you have a very analogous hierarchy of basic symbols, in the case of letters of DNA, building up to codons for amino acids, building up to proteins, and then building up to cells, tissues, and entire organisms. The extremely exciting thing is that many of the generative AI technologies that are being developed for the top row, for working with natural language, can be applied, in some cases, just right out of the box with no changes to biological language, or with relatively small changes and tweaks to accommodate the real differences that exist between the two forms of language. The ESM protein folding model, for example, basically uses Google's BERT model, which is now a couple of years old, but basically uses that directly for modeling protein sequences. So a lot of analogies here. The analogies do break down in some places, though. So obviously, you could say we invented language, we co-evolved with language, our brains have co-evolved with it. We understand it really, really deeply. We're really good at it. Humans have been kind of the market leaders in natural language for 100,000 years or so. We released our William Shakespeare product 500 years ago. It still sells extremely well. We're great at natural language. We are not great at biology today. Biology is, to some extent, it's like an alien language that we discovered about 50 years ago. It's the product of billions of years of evolution, and we are really wrestling with how to unlock it and how to unlock these different mappings that exist at different levels down below. I think that's in some ways extremely interesting when it comes to AI, because as you think about AI applied to natural language, it is competing with humans that invented natural language and are extremely good at it. A young child can master human language. At least it feels that way when I'm arguing with my 5-year-old. But we are, in general, really good at natural language. We are not good at biology. It takes 30, 35 years to reach state of the art in understanding and being able to manipulate biology for a person. We've only been working on it for at a molecular level for 75 years. So while AI has had to compete with our extremely high level of competence around natural language, it actually doesn't have to compete with that in biology. In biology, our human capability is way lower. The complexity of the substrate is considerably higher. And we only have a 50-year head start over AI in biology, not a 100,000-year head start like we do in natural language. AI is designed to be able to handle complexity really well, complexity that we've never really had to think through in biology. And so I think the potential here is for AI to move really fast in surpassing the state of the art as it comes to designing and working with biology. Advanced in a responsible and mature way, I think that is an extremely exciting opportunity. We at Ginkgo have been using AI and machine learning for a number of years, as you might imagine, in a range of different contexts. We've contributed to review articles on the topic. We've used it for biosecurity. You can look up our ENDAR project with the US government, and we've been using it in enzyme engineering, which I'll come back to later. So it's something we've spent a lot of time thinking about and adopting. I just want to leave you with in this part of the talk with kind of a very simple model of what AI can do for biotech. So at the top row of this diagram is what happens today. So a customer comes to us with a particular molecule they would like to make or discover. Then Ginkgo will run a series of large experiments, large physical experiments in the lab in order to collect relevant data sets and develop whatever the customer's product of interest is. At each step in that process, we'll leverage our existing knowledge and genetic assets in the form of code base, and together, hopefully that will help us get to the customer deliverable. What we are seeing with AI and what we think will accelerate over the next couple of years, given all of the most recent advancements in AI, is that first, that timeline is going to get compressed, and now down on the bottom row. So the time from the customer coming to Ginkgo with a particular ask to us being able to deliver to the customer, them being able to launch their product, hopefully, has gotten shorter, and will get shorter. It's getting shorter for a few reasons. The experiments are smaller. The experiments, we don't need to run as many of them in series, so it's faster. And all that happens by leveraging now not just code base, but code base plus AI. So we see this phenomenon happening already, and we think that we're really only scratching the surface of what it can do and how fast we can go and how cheap we can go. I'll recall the mission of Ginkgo: make biology easier to engineer. If AI allows us to go faster and cheaper, and as we'll talk about in a minute, perhaps getting better outcomes, then it is really helping us to achieve our mission. Okay, so a brief case study. This is probably kind of the deepest I'm going to go in the discussion today. Again, I just want to draw an analogy between natural language and biological language. So this stack, this kind of relatively crude stack that I'm showing on the left here, the Gen AI folks will know that I'm taking a bunch of simplifications here. But the basic stack is you have some large data source, some large corpus of text. You build a foundation model on top of that. You pre-train that on the language. And then you use a variety of techniques, whether it be fine-tuning or now RAG, retrieval of information from particular documents, to do task-specific things. So essentially, you can build a bunch of apps on top of your foundation model. So the foundation model can be something like PAM2. An app can be something like Bard or Duet AI, as we were hearing earlier. And you can build a whole bunch of apps on top of a single foundation model. That's the exciting thing, and for anybody who thinks about platforms, that's wildly exciting. On the right, I used Bard to help me. I put together a presentation on protein design at Ginkgo. Did a pretty nice job. As Scott was saying, these things can really shorten the time frames for typical business operations. Okay, let's flip over to proteins now. And enzymes, and look at a very similar stack. The underlying data now is not text, but it's sequence and structure information in a database like the Protein Data Bank. Sitting on top of that is some kind of large foundation model. AlphaFold, could be ESM, could be something new in the future. No doubt will be something new in the future. And then on top of that foundation model, you can build apps for designing protein sequences, predicting protein structures, looking at interactions between protein-protein interactions, protein-ligand interactions. A wide variety of different apps can be built on top of that foundation model. And again, as I think everybody knows, the performance of these foundation models, fine-tuned for particular applications, has been truly state-of-the-art and has unlocked incredible progress already in being able to work with biology and proteins. These are some of the predictions from AlphaFold here on the right. We've gone over the last 5 years from being kind of semi-stuck working at the level of linear sequences of proteins to now having a structure for basically any protein sequence that you can find, and being able to work at the structure level instead of constrained to working at the individual sequence level. And that's true across hundreds of millions of proteins in public and private databases. And that's just really an incredible unlock due to generative AI. Okay, same stack, now in a Ginkgo context. Now the underlying data in this more specific use case of enzyme engineering is that large metagenomic sequence database I was talking about, plus all of the enzyme screening data that I was talking about. Then we build our own AI models on top of that. We give them a cute name, OWL in our case, and then we build specific apps for either discovering enzymes with improved activity or improved specificity, stability, whatever might be the need for a given project. OWL, by the way, is comprised of a range of different AI and ML techniques, kind of indicated on the right here. Scott was talking about a co-pilot, a partner. I think that's very much how we see AI being used in practice in our projects, where our scientists, our protein engineers, use their intuition partnered with a variety of different AI models and apps in order to make fast progress. So let me show you an example of that. So this table describes the process of engineering enzymes at Ginkgo. So just to orient you, each column corresponds to essentially a round of engineering, a generation of engineering. So here there's 4 rounds of designing, building, and testing. And the rows in each column tell you a little bit about the specifics of what happens in each of those rounds of designing, building, and testing. So the top row tells you how many different unique designs we're looking at in each round. So it's thousands, typically. In the last round, we're down to just 100. Why are we down to just 100? We're down to 100 because of the quality of the AI ML approaches that we now have access to. Smaller experiments, cheaper experiments, faster experiments, better outcomes for our partners. I'll also draw your attention to the third row, experimental screening methods. That gets pretty in the weeds, but the thing I want you to take away from that is that at each of these rounds of, or each of these generations of engineering, we have to collect multimodal data sets and integrate those together in order to design the next round of variants or designs for the next generation. Lastly, I want to draw your attention to the bottom row, where we look at the improvement. So fold improvement over wild type. How much better did we get? First round, 1.5 times better, 3.9 times better. By the last round, we're 10 times better. In enzyme space, 10 times better than your starting point is often extremely good. Sometimes we do better than that. It depends on the particular problem. But what I want you to take away from this is that the AI approaches that are being used in the design step here allow us to make these generations or these cycles of engineering smaller and require less of them. So we don't have to go to generation 5, 6, 7, and 8 in order to get to the target performance like we would have had to do before. And that's very exciting and has real value for us and our partners. Okay, so faster, cheaper, Barry, is that all? Or can it be better too? Because that would be good. And so here's data from the exact same set of campaigns that we were just looking at on the prior slide. And here we're comparing a number of different design approaches. The OWL approach is Ginkgo's existing AI-ML approach, and then 2 conventional approaches in green and blue. And if you haven't seen these kind of plots before, it's pretty simple. Just the further up on the y-axis, the area or the kind of the center of gravity of the plot is, and how high the very kind of highest data points are, the better. So what we're seeing here is these AI-based approaches are outperforming classical and conventional approaches. So it can be faster, it can be cheaper, and it can be better too, and better outcomes. And in biology, it's often hard to get the right outcome. The probability of technical success is not 100%. So anytime we can get a better outcome like this, better performance, that has real value. Okay, so that was a little bit about using AI in the protein and enzyme engineering space. What I want to lay out is that for Ginkgo and for the industry as a whole, there really needs to be a roadmap here where you start with your models today, you collect additional large data sets, and then you build new foundation models and new apps on top of those. And so you could start at the protein level like Ginkgo has done, and then move on to the nucleotide level. That's something we're working on very actively internally at the moment, so that you have a DNA or an RNA foundation model and applications built, specific applications built on top of that. And ultimately what we want to do is we want to build whole cell models. We want to be able to predict the phenotype of a cell when you change a piece of DNA or when you expose it to a drug compound, for example. We want to be able to model the behavior of cells so that we can have advanced cell therapies that can permanently cure a disease. The language models that we're using today, the transformer-based architecture, is proving hugely useful to address some of these challenges and helping us move way further towards the right to higher level, more complex models. I think I would speculate that in order to be able to truly model cells and tissues, which is what to some extent we want to be able to do in the long run, we're going to need new approaches. We're going to need new types of computation. We're going to need quantum. We're going to need more scale. And we're excited to be on that journey with Google and with our partners and with all of you in the broader community. So that's it for me today. I'm going to leave it. Just please remember, DNA is code. It was invented once. Our platform, powered with AI and partnered with Google, is making it easier to program that DNA across a range of markets. Please contact us. My colleague Al at Ginkgo Bioworks is getting a lot of emails at the moment, but please email ai@ginkgobioworks.com. And I'll leave it there. Thank you. And if I'm happy to take questions if there's time, or I'll be around a little bit if that's easier. Thank you. Thank you.
Any questions for Barry? This one. Yep, there we go.
Really interesting, thank you. I was curious, you talked about the different types of data that you guys work with or generally are worked with, and I was wondering which ones you think are being underutilized? Like which types of data in this space do you think might yield solutions in the future that are untapped?
Yeah, super interesting question. I would say that we have gotten, over the last 20-odd years, we've gotten very good at working with nucleotide data, whether it be DNA or RNA, and it has become extremely easy to collect that data. I think what we're very excited about is the things happening at the cellular level, the use of high content imaging to capture images of entire cells, populations of thousands of cells, and use that to understand and learn more about the behavior of single cells, but at the cellular level. So another trend that you may be familiar with is a migration from population level data, where you're taking some aggregated measure of the behavior of 1 million or 1 billion cells, and now being able to zoom all the way down to individual cells and collect all kinds of omics data, whether it be genomics, metabolomic, RNA data, on proteomic data on individual cells, to understand the kind of the diversity of behavior at the individual cell level, and that's unlocking a lot for the industry.
Thank you. Great presentation. Another data-related question. You show that slide with the requirement or benefits of using multimodal data with lots of different sort of analytical techniques,
Yep.
mass spec, NGS.
Yeah.
Is that data readily available and accessible and prepared for the use cases that involve AI, or are there challenges in assembling that data or getting at that data?
Yeah, there's I guess there's like a public context version of that question, and there's like inside a place like Ginkgo. We have spent a lot of time building centralized data lakes that store all of that different multimodal data across projects, with all the appropriate linking to the underlying DNA sequences and cells from which the data came. So we've done a lot of work on that area. We still have a way to go in order to make it trivial for data that we collect in a particular project to just flow directly into model training. There is still, cleaning and curation work that has to happen, data engineering work that has to happen. But certainly our goal is to just see that be a very straightforward flow. If you're looking in the public context, you're a researcher, you want to be able to broadly access multimodal data. I think the picture is a lot harder. You're going across different databases. There's missing metadata. There's metadata that doesn't agree. There's a reasonable amount of agreement and standardization on data formats. And then there is also then just large gaps in data sets that are not publicly available. That's a reason, I think that in part, that is a reason why a lot of the early AI work has happened in the protein space, because protein sequences and protein structures, the publicly available data is actually relatively good and at scale. But you go outside of that, outside of DNA and protein sequence level information, and it gets much spottier, I would say.
Hey, wanted to harken back onto one of the comments that you made on biology is easy to program but hard to predict. I mean, it's wonderful to see all these models being applied to biological problems. But one thing that's still hard to get by these days is insights and explainability or interpretability of these models, especially when it comes to models or language that biology is speaking. And Ginkgo is well positioned, given the vast amounts of data that it has in-house to understand mechanisms that might actually compress many of the inputs that would be needed. So I was just wondering if you could speak to how you would leverage some of these models to maybe derive insights, explainability, insofar as in biology.
So there's probably a couple of levels to that question. So first of all, data, high quality data being used to train best-in-class models today is answering biological questions directly. For us and for the industry, and that's amazing. The models are somewhat of a black box, and they're also very large. There are interesting studies emerging about the interpretability of the model, looking in at individual layers, looking at particular attention heads, and mapping what those attention heads are doing to a particular biological mechanism, for example. And that's very interesting because I think there's the potential there for us to learn new biology by interpreting what the model is figuring out. It seems like we are still at a very early stage of that. But I think it's pretty exciting. The other potential benefit of that is maybe we can make the models smaller by injecting some degree of human understanding, some degree of mechanistic modeling as well, so that training and inference can be faster. But it feels early days in terms of being able to interpret the models. Right now, I think we're all exploring just what are the range of problems they can answer for us directly. But the interpretability question is very interesting. Okay, thanks everybody.